those of you who don't know, my name is Kristen Thomas, and I'm one of your Frontiers coordinators for this season. And today I have the honor of introducing our speaker, Dr. Lary Walker, from right here at Emory. Dr. Walker got his PhD from Emory University before beginning postdoctoral research at Emory, then at Johns Hopkins. Then he joined the faculty at Johns Hopkins in the Department of Pathology until 1995, where he studied senile plaques and cerebral amyloid beta angiopathy in humans and non-human primates. In 1995, he moved to Park Davis, Orlando, to study ground-like induction of amyloid beta amyloidosis in transgenic mouse models. In 2003, he returned to Emory to study the mechanism whereby Alzheimer-associated proteins, amyloid beta and tau, form pathogenic assemblies in vivo, and how these agents are trafficked within the body. Today, he will be discussing current knowledge of the seeded induction of protein aggregation, and how this may explain the onset and progression of multiple neurodegenerative diseases, specifically including Alzheimer's. So, please join me in welcoming Dr. Lary Walker.
Thanks, Kristen. Definitely including Alzheimer's disease. I'll get to it eventually, but many of you have heard bits and pieces of the seeding story before, so what I'll do today is take a more conceptual approach to the whole issue of protein seeding and describe how protein seeding, I think, is involved in a number of different diseases, and I think as a concept developed in an extremely interesting way. So I'm going to take a historical trajectory on this, so to speak, just to give you an idea of where this whole idea came from. So just as an overview of what I'll discuss, first very briefly just talk about proteins and the prion paradigm for protein seeding. Then I'll spend a little time talking about our studies on the initiation of the aggregation of A beta in the brain in a prion-like fashion. And then just briefly discuss how other diseases may involve this very mechanism. And then finally discuss some of the therapeutic and theoretical implications of this concept. So as you all sit there, of course you're eating, what you're eating is proteins. Proteins are broken down into amino acids, and the amino acids then are restrung together by your cells. The cells are just turning out proteins at an extremely high rate all the time. And they're made essentially in a string of amino acids, each of which is unique to a particular protein. And since the amino acids all have particular characteristics, chemical, physical characteristics, they tend to either attract or repel one another in different ways. So most of the proteins that you make, right after the amino acid sequence is formed, these proteins will snap into a particular configuration. And this shows the three main structures that a particular protein might have. So this represents an amino acid sequence. They can form a helical structure known as the alpha helix. They can form beta strands, or they can be random coils, sort of floppy segments of proteins that have no regular structure. So this is what makes the proteins functional. In most cases, their structure determines how they're able to behave in the body. So the proteins are really sort of the engine and the chassis of life. They're enzymes, they're structural, they have all sorts of functions. And these functions depend, in many cases, on how they fold. So the question becomes, what happens when these proteins misfold? In other words, what happens when they fold into a different shape? And this is the theme of today's talk. And what I'll argue is that this misfolding, although remarkably common, also is a common source of many of the diseases of old age, including Alzheimer's disease. And the story begins with the bizarre biology of one of the most controversial molecules in the history of medicine, and that is the prion. And the prion story begins actually in the 1700s with a disease of sheep called scrapie. called scrapie in Britain because, as you can see, the animals have a tendency to rub against whatever surface they can find. This rubs off the wool, which of course is a problem if you're a farmer interested in wool. But more seriously, these animals eventually develop motor problems, coordination problems, and within 3 to 6 months they die. This was actually endemic in many parts of England from the 1700s on. It was well known to farmers, and it was a serious problem, but within a given flock, maybe 1 to 2% of the animals would be affected. When pathologists looked at the brains of animals that had scrapie, they noticed some very interesting findings and very unusual findings from a neuropathological standpoint. One obvious change is holes, or better stated, vacuoles. These large bubble-like structures, which have given these diseases one of their many names, and that is the spongiform encephalopathies, because the brain actually resembles a sponge. So you have all of these holes, which pathologists are sometimes reluctant to make a big deal about, because holes can happen for lots of artifactual reasons. And so there have been instances where they've been seen in the brain, but not recognized for the importance that they have in particular diseases. So that's one thing that was unusual in the brains of the scrapie sheep. Another was the proliferation of activated astrocytes, astrocytosis. This, of course, is an immunostained section, so this was done much later than the original findings. But the astrocytosis was quite apparent to the pathologists who were studying this. What's more, the brain showed no obvious evidence of an inflammatory response. So if a bacterium or a virus was causing this disease, you would expect to see all sorts of changes, not only in the brain, but elsewhere in the body. There would be an increase in temperature and all of these sort of things. No evidence whatsoever of this in scrapie. It was also known by the farmers, really since the 1700s, that this seemed to be infectious. In other words, a flock that had scrapie tended to pass that problem on to subsequent generations, and it was very difficult to rid a particular field of scrapie in the sheep. And in the 1930s, a French group and a British group more or less simultaneously showed that scrapie could be transmitted by inoculation of a sheep with brain material from sheep that were afflicted with the disease. And this was not terribly surprising, because they kind of suspected that this might be an infectious disease, but it was a little surprising because it didn't seem to involve bacteria or viruses. And what's more, it took a long time for this to happen. When you're infected with a bacterium or a virus, things usually take place fairly quickly. In this instance, it took 3 months, 6 months, sometimes even longer than that for the disease to appear. So it looked like it was infectious. It was a very interesting disease, but this was something that really was only known to farmers, mostly in England, and to the veterinary pathologists. It was more or less confined in terms of the knowledge of it to these groups. This began to change in the 1950s with studies conducted by Carlton Gajdusek, a brilliant and troubled scientist, his colleague Vince Zekas, and others. Gajdusek was working in Australia at this time, and he heard rumors of a strange disease in New Guinea that was called kuru. And being a peripatetic sort of person and one who really was extremely impatient, he headed for New Guinea to learn as much as he could about this disease. And what he found was that this was confined to a particular tribe, the Foray tribe in New Guinea. And it was also mainly confined to children and women within the Foray tribe. Unusual, not suggestive of an infectious disease, or even, in a dubious sense, of a genetic disease. And so they had no idea what was causing it. And the disease began with cognitive changes, motor changes. One of the characteristics of kuru was sort of a bizarre pathological form of laughter which led the Australian press to refer to this as the laughing death, because within 3 to 6 months, usually, those who contracted the disease died. It was invariably fatal, as was Kuru, or as was Scrapie. So these guys were trying to figure out what was happening in the case of Kuru. And this is one of my favorite pictures in the annals of the history of medicine, because here you see Gajdusek looking into the microscope. Here's Vince Zegas. Here's Jack Baker, who was the patrol officer in New Guinea at this time. And they're all basically trying to figure out what's happening to Kuru victims. And here on the table is the brain of a victim of Kuru. And this is Jack Baker's dining room table. And it was on his table that they did the autopsies for this disease. So as you might imagine, this was a different time. So they were doing all these studies. They were looking at the brain. They were looking at all the organs they could to try to figure out what was going on. And the one thing that they did, they sent the brain to a pathologist in Washington, D.C. named Igor Klatzo. And Klatzo looked at the brain and said, God, this is really bizarre. And he happened, however, to be aware of a very rare human disease at that time, almost unknown, called Creutzfeldt-Jakob disease. And when Klatzo looked at the brain of the Kuru patients, he said, well, what I see are holes and astrocytosis. And the closest thing I can think of in human diseases to this is Creutzfeldt-Jakob disease. So this was interesting and important, but again, Creutzfeldt-Jakob was practically unknown at the time. No one quite knew what to do with it. But Gajdusek, being a peripatetic sort of person, put all of this information together. He had made movies of the Kuru victims. He had all sorts of pictures of tissues and whatnot, pictures of the tribesmen, and he put together a traveling show. And he went from museum to museum around the world with this show, just trying to explain this very bizarre disease, the likes of which no one had ever seen before. And at one point, the show stopped at the Wellcome Medical Museum in London. And it happened that a friend of this guy, Bill Hadlow, was traveling through London at the time. And Hadlow was an American veterinary pathologist who was working at the Compton Research Center just near London on scrapie. And he was sent by the U.S. government to this location to study scrapie because there was virtually no research on scrapie being done in the U.S. It really wasn't considered to be that big of a problem in the United States. So he went to England where the expertise and the experts were. This friend came and had dinner at Hadlow's house one night and said, so Bill, I was at the Wellcome Medical Museum recently, and there's just an interesting exhibit about a strange disease of a tribe in New Guinea that I think you might be interested in. And Hadlow was interested. So within the next week or two, Hadlow made his way into London and checked out the exhibit. And the first thing he noticed was that the pathology that was taking place, that was happening in the brains of the Kuru patients, was identical. He was stunned by how similar it was to the pathology that one sees in scrapie. So Hadlow then communicated a little bit with Gajdusek. He wrote and asked for some reprints. Gajdusek sent him, and Hadlow became more and more agitated about this because he knew that he was onto something. So he wrote a letter to The Lancet, which is, of course, one of the premier British medical journals, describing the similarities between Kuru and scrapie. It was a very brief letter. At the time, there was a printing strike going on in England, so in order to speed the information to Gajdusek, he went ahead and sent the letter to Gajdusek as well. And basically, the letter pointed out these similarities. The similarities not only in terms of the pathology, but in the course of the disease and all of the other strange characteristics that these two diseases had. So Gajdusek got the letter, and what does a good scientist do but bluff? And he wrote back, and there's no doubt he was stunned when he got this, because he had pretty much written off the idea that Kuru might be an infectious disease. So he got this letter, and in the letter to Lancet, Hadlow said, well, so since there are all these similarities, and since we know that scrapie can be transmitted from one sheep to another by inoculation of brain material, it might make sense to try inoculating Kuru brain material into non-human primates, which might be susceptible to this, and which might show that it's an infectious disease. And Gajdusek wrote back and said, oh yes, we've already begun these studies and blah, blah, blah. Gajdusek himself actually confessed several years ago, well, no, I really haven't. In fact, it was about 4 years later that they began the studies of infectivity in non-human primates. And because this disease takes a long time to develop, it took a long time for the studies eventually to lead to publications. But beginning in the late 60s, the Gajdusek group began publishing the results of these studies, and they were clear and unequivocal that Kuru is infectious in non-human primates. Not only that, because they were aware at this point of the connection with Creutzfeldt-Jakob disease, which occurs spontaneously around the world in 1 in a million people. Now, no one to this day is sure why this happens in this way, but they did notice very early on that Kuru and Creutzfeldt-Jakob disease did have this in common. And Gajdusek showed not only was Kuru transmissible, but Creutzfeldt-Jakob disease was as well in a very similar way. This then, one thing led to another, and in the 1980s, Stan Prusiner at the University of California, San Francisco, began publishing, well, actually he'd been publishing since the 70s, he'd been working in the field since then, but Prusiner coined the term prion, because his work had shown that it appeared not to be a virus, bacterium, any normal infectious agent, but in fact was simply a misfolded form of a protein that's causing this disease. And this created a firestorm. There were all sorts of people who said, oh, geez, what's this guy coining this new term, and blah, blah, blah. But the term has stuck, and I think the concept has stuck as well. And in fact, Gajdusek won the Nobel Prize for his work on Kuru in the 1970s, and Prusiner won the Nobel Prize for his work showing that the infectious agent is a protein in the 1990s. So what are the prion diseases? I've mentioned Creutzfeldt-Jakob and Kuru. There are other extremely rare genetic forms of prion disease in humans. And in fact, the diseases in humans tend all to be, or mostly to be either idiopathic or genetic in origin. The non-human prionoses, such as scrapie, are another story. In fact, there is a form of prion disease in the United States and Canada primarily, the western states, called chronic wasting disease, that is transmitted from deer to deer and deer to elk and among servants, basically. And it's spreading like wildfire in western North America. Fortunately, neither scrapie nor chronic wasting disease has shown any convincing evidence of being transmissible to humans. In other words, scrapie has been endemic in Britain since the 1700s, and there's been no evidence that this has led to an increase in the amount of Creutzfeldt-Jakob disease in Britain, which one would expect if indeed there was transmission.
Larry?
Yes?
How exactly is it transmitted from animals?
Well, the chronic wasting disease, actually these tend not to be, most of the animal prionoses involve humans in some way. In other words, they're either farmed animals or animals that have been infected, as I'll talk about with mad cow disease. The chronic wasting disease, they think it's either being transmitted by the animals consuming infected feces or possibly urine, or nose-to-nose contact, or a nose touches a fence or a tree and then another animal touches the same tree. They're not really sure, and it may be all of those sorts of things. But it's a very important question, and it indicates one of the important characteristics of prions, and that is they're tough. They're aggregates of protein that are extremely difficult to get rid of once they form. So the best known, when I talk about this usually, especially to lay groups, and I say, here are prion diseases or Creutzfeldt-Jakob and then a mad cow disease, oh yeah. And mad cow disease became extremely high profile in the 1990s because it was shown then that this developed in Great Britain beginning probably in the 1980s. And it's thought that what happened was that meat and bone meal, which was fed to cows to supplement their food, was made of animals that had died. Many of these animals were sheep. Some of the sheep, of course, had scrapie, and it's thought that in 1 or 2 cows, this was transmitted to the cow through the meat and bone meal, and then it developed sort of a chain reaction. The infected cows were fed into the system, and more animals got it. And in the end, hundreds of thousands of cattle had to be slaughtered in Britain in order to stem the tide of bovine spongiform encephalopathy, mad cow disease. And it was successful. The problem has almost disappeared in Great Britain. It was also a bit of an issue in some other European and other countries. But by eventually realizing what was happening, they were able to stop this disease. But of course, mad cow became an issue because despite early denials, it was eventually recognized that in some cases, humans were coming down with previously unknown form of Creutzfeldt-Jakob disease from the consumption of meat that was tainted by mad cow, the prion protein. And to this day, I think there have been approximately 200 cases, human cases, of this variant Creutzfeldt-Jakob disease that have been described. And the epidemic in humans, fortunately, has peaked. Now there's maybe 1 or 2 cases a year that are appearing, but it appears that the problem is over. But what all of this shows is another heretofore unknown characteristic of any disease, and that is that these are caused by unknown mechanisms, the prion diseases. They're caused by genetic changes in the prion protein, and there are many mutations that can cause prion diseases. And they can be infectious in some circumstances. In humans it's unusual, in animals not, but infectivity is definitely an issue. So what's causing the infectivity? Well, here we go back to the protein. So this is a hypothetical model of a prion protein as it normally folds. What is happening in prion disease is that the protein is adopting a lot more of this beta strand, beta sheet conformation. And this new conformation causes these molecules to be stable, and it causes them to be sticky. They bind to one another, and when they bind to one another, if this molecule binds to a normal molecule, it will corrupt the normal molecule and cause it to assume this shape. In this conformation, they stick to one another, they form larger and larger aggregates, and eventually the fibrils that they form bundle into these aggregates of protein that are associated with the development of prion disease, these so-called amyloid aggregates of protein. So I'm going to show this one time. This is just my animated model of what's happening in prion disease. As is the case for all diseases that cause these neurodegenerative diseases, we all make these proteins normally. We think what's happening is that they misfold, and proteins misfold all the time. The cells generally can get rid of them. For some reason, these misfolded proteins escape the cellular maintenance mechanisms and persist and are able to propagate their misfolded characteristics. So what happens is the prion protein misfolds. This protein then encounters a normally folded version of the prion protein and converts it. These are sticky, they aggregate into bigger and bigger structures. These are still at the ultrastructural level. And at the light microscopic level, we get these amyloid deposits of protein. These can fragment, and the fragments have ends that themselves can convert prion proteins. So you get a growth and a spread of these toxic molecules that can corrupt other molecules. And although this is what we see with the light microscope, there is increasing evidence with prion disease and Alzheimer's disease that in fact the elements that are most destructive to cells in the brain are the oligomers, the small ones that can float about and that have per number of molecules the largest number of cases that are capable of propagating this disease. Okay, is that all clear? Do you have a good idea of what's going on? So let's then go to Alzheimer's disease. Alzheimer's, you all know, I won't spend a lot of time on it, but it was named after Alois Alzheimer, who just a little more than a century ago studied a patient known as August D. Alzheimer didn't describe dementia for the first time. Alzheimer's is the most common form of dementia. It affects 5 million Americans. The number is expected to go up very rapidly as our population ages. He didn't describe dementia for the first time, but rather he related dementia to the light microscopic appearance of these two lesions. And this is a silver-stained section of the brain of an Alzheimer patient, similar to what Alzheimer would have seen himself. And he saw the senile plaques, which are the black structures here, which are accumulations of the extracellular protein A beta, and neurofibrillary tangles, which are intraneuronal accumulations of the microtubule-associated protein tau. These are absolutely characteristic of Alzheimer's disease, and to this day they're used to diagnose Alzheimer's disease in patients who are demented. We think that the aggregation of A beta is the primary problem in Alzheimer's disease. And we think that for a number of reasons, but one is that in cases of Alzheimer's disease, if the brain has senile plaques, the brain also has neurofibrillary tangles. So they always are related in Alzheimer's disease. There are, however, cases of primary tauopathy where the tau gene is mutated and the patient develops nothing but neurofibrillary tangles. And in this case, the tangles fill the brain, the patients develop all sorts of cognitive behavioral problems, but senile plaques do not appear. So it looks like it's a one-way street from A beta aggregation to tauopathy. What's more, every genetic or environmental risk factor for Alzheimer's disease influences the metabolism of A beta in some way or another. So there is little question that A beta is a central and essential component of the cascade that leads to Alzheimer's disease. So A beta itself is, again, we're all producing this fragment. It's a 40 or 42 amino acid segment of the A beta precursor protein known as A3P. It's cleaved from the precursor protein, which spans the membrane one time. And if it's cleaved in its entirety, this version tends to misfold and tends to be sticky. And this is why it's related to the disease. There is some evidence that A beta may have some physiological function, may be involved in learning and memory in some way. But what we do know is that when it accumulates to levels above physiological, this leads to an increased risk of developing Alzheimer's disease. So this takes us back to prion. It had not escaped the notice of scientists who were studying prion disease and pathologists that there are some similarities between prion diseases and Alzheimer's disease. In this case, the proteins involved, prion protein or A beta, are stained, immunostained with antibodies to the respective proteins. And you can see that there had been speculation that maybe these two diseases have something in common. They both involve the slow, steady degeneration of the brain. They both involve the accumulation of particular proteins. You don't see usually spongiform change in Alzheimer's disease. But still, the similarities were enough that, well, maybe they're the same. So if that's so, then maybe Alzheimer's disease might be inducible, might be, in a sense, transmissible, like prion disease is. So this was tried by the Gadjusek group beginning in the late 60s and by a group in Britain. And the Gadjusek group ultimately concluded that no, there wasn't really good evidence that they were seeing transmission of Alzheimer's disease. But it's a long story, and the study had lots of flaws. We could talk about it later. But what's important is that the British group did find evidence that the number of senile plaques in marmosets is much greater in those animals that had received Alzheimer brain extracts than those that had received control injections. Alzheimer brain homogenates, actually, in this case. However, it took 5 to 6 years before increased plaques were evident in the marmosets. And so this was never replicated and really didn't receive a whole lot of attention. And this is where my lab came into the story. In the mid-1990s, transgenic mouse models of Alzheimer's disease appeared. These expressed the human amyloid A-beta precursor protein, and usually at fairly high levels. And normally, as they age, they will develop senile plaques, here shown in the silver stain, all these black blobs in the cortex, that increase in number as they age. Usually, this particular model doesn't develop the plaques until 9 months of age. And this gives us a nice window within which we can determine whether we can accelerate this process in a prion-like fashion, that is, by injecting Alzheimer brain extract. So what we did was essentially what the folks who did prion studies did. We took A-beta-rich material from the brains of Alzheimer patients and homogenized it, spun it in a centrifuge, and injected the supernatant into transgenic mice. And then we waited varying periods of time to see what happened. And we sectioned the brain, in this case after 5 months, and this is what we see. On the injected side, the side that received the Alzheimer brain extract, there's a significant increase in the amount of A-beta in the hippocampus. And we usually do unilateral hippocampal injections in this case. On the other side, where you inject, say, a non-Alzheimer brain, PBS, nothing at all, whatever, there is always much less, if any, A-beta deposition. So this indicated that the Alzheimer brain can stimulate this process in transgenic mice. But what's the evidence that A-beta itself is the so-called transmissible agent? Well, first of all, we know that it's not the injectate itself. You might imagine that the injectate would be immunoreactive. Because if we look within the first month after the injection, we don't see anything in the injected hippocampus. Rather, the amount of A-beta in the hippocampus increases over time. So we're stimulating a process. The donor brain extract must contain A-beta. So if we inject a control human brain extract, a non-transgenic mouse brain extract, whatever, this will not result in seeding within the same time frame as the Alzheimer brain extract might. The host brain must express human APP. So mice and rats normally make A-beta, but it has 3 amino acid differences that make mice and rats resistant to the formation of plaques. So they have to be transgenic to be able to do this. If they're not transgenic, nothing happens when the Alzheimer brain extract is injected. And finally, if we